Okay, so today we are going to be talking about analyzing structure and function of genes. I'm going to be primarily using the Albert's book for this, and this is chapter 10, which is something that you guys had asked for. But this will also work for the genetics folks because a lot of this material is also stuff that we cover in genetics as well. Okay, so let's go ahead and get started. So today our basic kind of outline for this particular lecture, we're going to be talking about how we isolate and clone specific sections of our DNA molecules. So maybe a gene of interest or a little fragment of DNA that we're interested in learning more about. We're also going to be talking about how we can utilize polymerase chain reaction or PCR for cloning purposes. And then we are going to talk about different methods and applications of sequencing the DNA and how we can learn about a gene's function once we have its sequence or once we know about its existence. So I can't find my cursor, sorry. Okay, so let's start off by looking at how we can isolate and clone parts of DNA. Usually it's in the shape of a particular gene of interest, particular sequence of DNA. So this is a big application for this would be how we are able to selectively breed or create a particular plant or animal the way we want with specific characteristics. Now you don't necessarily have to know the sequence of that particular gene or even anything about that gene as such. A lot of the initial selective breeding happened just because of selecting individuals that had those characteristics, whether it was a plant or an animal that we wanted to have in the next generation and have, selectively breeding them in that way. So that is a form of genetic manipulation as well, right? Because over time you can artificially increase the alleles, the frequencies of those particular characteristics that we find favorable for whatever reason. However, another way to do this type of genetic manipulation is to actually introduce the actual gene sequence and make those changes that we want to make in there as well. So to do that, we would need to know the actual DNA sequence of that particular gene. So just to kind of go back and review some of the DNA basics, if you remember, each person has a unique DNA code, unique genetic code, except for identical twins that will share the same thing. It serves as the blueprint for each individual, and it's composed of 4 nucleotides, the ATCG, right? That's what they're usually written as. And those would be what you'll see in the DNA structure. 99.8% of the sequences are shared amongst all human beings, but then there are those changes that make us who we are based on the different alleles that are present within an individual. Now, just to remind yourself of some other stuff, one of the things that we are going to be using to take portions of DNA so we can learn about them more are a special type of proteins called enzymes. Enzymes are biological catalysts that are found in all cells. They are usually proteins, and their purpose is to speed up chemical reactions. So they're not actually causing the reaction to occur, they're just speeding up the reaction that was going to happen. They don't get used up in the process, so they can just go through and do that cycle over and over and over again. So they're recyclable, they are reusable. They have a very unique structure. Each enzyme has its own unique structure so that they can only work on a very particular substrate. So it makes them very specific to a particular reaction or part of reaction. And because they have to maintain this very specific structure in order to work appropriately, they work optimally under a very specific environmental condition. It will be different for each enzyme, but it's going to be based on the important portion or the active portion of that enzyme, maintaining that chemical structure so that it can perform the work that it needs to do. And because proteins can be sensitive to both temperature and pH changes, it usually means that the temperature and the pH has to be within that optimal range. They are extremely important for all metabolic processes inside our body. Many of the reactions that happen in our body cannot happen at room temperature or at physiological temperature without the help of enzymes. They usually require a lot higher temperature to work best. But obviously, we can't have that inside our body, or the body would cease to exist. So instead, our bodies have enzymes, or our cells have enzymes that take over to help these reactions, to speed them up, to allow them to happen under physiological conditions. So this obviously includes metabolic processes like digestion, protein synthesis, but also DNA replication and processing as well. So we're going to be talking about a specific class of enzymes called restriction enzyme. Restriction enzymes have lots of functions inside the body, inside the cells, but they also have many, they are very useful. They have many functions outside in research labs as well. They are the key force that allow us to make recombinant DNA and to do cloning of specific portions of DNA so that we can selectively create a particular type of plant or animal or another structure that we find useful. This is how a lot of the GMOs are made, right? The genetically modified organisms, they rely on this type of technology. They can also be used to create DNA profile analyses. We use restriction enzymes and use the patterns that are created by them in each individual to diagnose diseases, to look for paternity or family relationship, in forensics to identify the victim or to identify the culprit, or to tell us if our, or to kind of make sure that we have the right culprit as well. So a lot of times it's used to exonerate people of the crime that they are being accused of. So restriction enzymes, the way they work, and we'll talk about that in a second. These enzymes were first discovered in bacteria. They help the bacteria destroy, it's a protective mechanism. They help the bacteria destroy viral DNA through this mechanism. They destroy that so that they cannot be infected by it and they cannot be destroyed by it themselves. Restriction enzymes come in many different flavors, many different types. Each one cuts between specific DNA bases, a sequence of double-stranded DNA, so that they can destroy that portion of DNA, or they can cause it to be broken at that point. Obviously, the more times a particular sequence is present inside a molecule, the more cuts will be made. And by doing that, you can cut the DNA into multiple pieces. You can also use a combination of enzymes to allow for multiple cuts rather than a single cut. These pieces can then be separated by gel electrophoresis, and you can monitor their size, their number based on the intensity, and the size that is listed based on how far they migrate in that matrix of gels that we create. So, looking at some examples for restriction enzymes can cut two different ways. at the end of the day. They can make up what we call a blunt cut, where it's just a straight cut on the double-strand DNA, and both sides are cut at the same point. Or it can create what we call sticky ends, in which it is a staggered cut, where in the recognition sequence that the enzyme recognizes, the cut is made in a way to have overhanging single-stranded DNA ends, which are termed sticky ends because they can be used to combine two DNA molecules together that were cut with the same restriction enzymes but may not have come from the same place. So for example, ECOR1, and this is what they are showing an example of, will cut after this first G in this particular sequence. So this sequence is a palindrome, so it's the same reading on the on both strands. One when you go from the 5' to the 3' direction, right? So because those strands are anti-parallel to each other, it's going to cut at the G, leading to this overhang or sticky end in front of it with the remaining sequence. And that can help us create different combinations that wouldn't have existed otherwise. So this was first discovered in E. coli bacteria. That's why it's ECO. So whenever you see the ECO R1, so that's ECO is from the E. coli bacteria. The resulting pieces of DNA, these little fragments, are restriction fragments. So, like I said, restriction enzyme will cleave both strands of the DNA, and they cleave at very specific nucleotide sequences, and that's unique to each restriction enzyme. So that if we are looking at multiple enzymes, you can identify where they're going to cut based on the sequence that they recognize. Here, for example, you have 3 different restriction enzymes, each with its own unique sequence. This Hey3 cuts in between, makes a blunt cut, so it makes a straight cut. In this 4 nucleotide sequence, ECOR1 and HIN3 both make sticky end, right, cuts. So those are not blunt cuts, they're sticky end cuts in a 6 nucleotide sequence, but that sequence is different. So in the first one, it's after the G that it's cut, and in the HIN3, it's after the A. But both of them lead to a sticky end that can be utilized for cloning purposes. Now, if you were to cut one of your DNA fragments, DNA molecules with ECOR1 and the other one with HIN3, you could not combine them together because the sticky ends, while sticky, are very different. They are not the same sequence in those two. But if you cut both of them by ECOR1 or HIN3, then you could use the sticky ends to combine the various molecules together and to create a new molecule that contains part of the fragment of each combined together. Okay, so here is an example of how that might work. So here you have, again, examples of protruding or sticky ends versus blunt ends. They are usually named based on what type of bacteria or organism they were found in. So HIN3 was discovered in H. influenza, PST1 in P. stauretii, and ECOR-V in E. coli. So ECOR we saw before too in ECOR1. This is ECOR5, and they are both discovered in E. coli. So the advantage of protruding ends are many. And so here is a question for you to ponder. If we had these sticky ends, why might these be useful? And I've given you some hints already about them, but you can think about it for a second as well before we talk about it some more. Okay. So before we go further, if we had enzyme, a DNA molecule that was cut with eCor, for example, eCor5, that creates blunt ends. With these blunt ends, if you try to combine it back together, there was no guarantee that you would get the correct orientation in the final molecule. Because it is a blunt end, it can attach back with itself, obviously, but if there were two different molecules present that were both cut with eCor5, the second molecule could flip either way because it's a blunt cut and still attach in here. That would lead to not exactly the same molecule that we want as the only option, and you would have some that would have self-sealed. You would have some that were in the correct orientation and some that were in the flipped orientation. However, if instead you had molecules that were cut by a protruding end producing enzyme, in that case, the only way that they could combine, as long as you had your sequences correct, the only way that they could stick back together would be for it to be in the correct orientation. So the protruding ends allow for the correct orientation to be the only possible outcome from your ligation reaction, other than the self-sealing. Of course, sometimes you can still get the self-sealing, but outside of it, the only way that you can get your cloned fragment in there would be in the correct orientation, because you would have accounted for that in your cuts. So there are many uses from restriction enzyme. Once we can cut the DNA, we can cut it up in the same organism, in the same cell. You can cut it up from different people and combine together. You can cut up different organisms' DNA and then combine them together too, or compare it. And you could use these different combinations for cloning purposes. You can use them for forensics. You can use it in medical diagnostic to see if a particular lethal allele or a particular harmful organism is present, infectious agent is present in the system or not, for paternity determination, and also to examine evolutionary relationships between various organisms across your family tree or across the life tree. The way to combine them together requires a ligation reaction, which inside our cells required another enzyme called a ligase. If we are producing these ourselves in vitro in a research lab setting, we still need a DNA ligase, but we can provide it to it artificially through our own mechanisms. So in our little tube, we would have the DNA fragments that we have created, the restriction fragments that we want to combine. We would provide it with appropriate energy source in the form of ADP. We would provide that with DNA ligase so it can actually do the ligation reaction, and then the right temperature and the right pH, just like usual, so that the reaction can occur. And you would also provide it with the NTPs to fill in the gaps if there are any sticky ends, and then you are going to allow the reaction to occur at the right temperature so that you can end up with the joining of the two strands and getting that final molecule. So here are two different examples. In the first one, you are joining two fragments that are cut by the same restriction enzyme that have produced protruding ends, so sticky ends. And when you combine them together, it takes ADP, some energy, and DNA ligase to work together to combine it together and kind of line up on top of each other and get joined together through that. You can also look at a possibility where you are joining two different fragments that are cut by different restriction enzymes. Maybe you were not able to find a perfect match, and you had needed a way for it to still combine given the biology that you were looking for. In that case, you could take one of them and cut it up with a protruding or sticky end, another one with a blunt end, or you can use two different enzymes that create blunt ends. And then this way you are gonna have to fill up this remaining sticky end first, which is gonna require that you also provide it with DNA polymerase and dNTPs so it can fill those in. And then now it's essentially a blunt cut, which will take the same way DNA ligase, ATP, and create your final new molecule. In this case, remember, it can be flipped, so you can have it in either orientation, and you would need to see what percentage of it or what part of it is your orientation. Now, in this case, the resulting molecule is going to end up losing that original restriction site in many cases, because these were two different NTPs, two different sequences that are now joining together, and part of the original sequence is going to be lost in the process. But regardless, this process allows us to clone a molecule, a gene, a piece of DNA that we are interested in, and look at it in a different way, to examine it, to study its function, to study its how it affects different organisms, and then also to clone it to create sometimes a new genetically modified organism of some sort, a plant or an animal or another organism, so that we can have it do the work that we want it to do. Maybe we are looking for a drug bug resistant crop of a particular grain, or we are looking for understanding the role of a newly identified gene. Either way, this is how we would use it to create or clone our DNA into some type of a vector, some type of a backbone to work with. This DNA backbone is usually a plasmid, a circular piece of DNA that we are inserting this clone in. Again, maybe I'm going to make sure that both the vector, this molecule that we're going to use as the backbone, as well as the insert, the DNA that we want to, the sequence of DNA that we want to put in there, are both cut with the same restriction enzyme, with matching sticky ends in that case, and then they are going to essentially lock and key into each other with the help of ligase to create the plasmid containing the gene of interest, or the DNA molecule of interest. So the purpose of this is to make recombinant DNA. So DNA fragments are, like I said, usually inserted into bacterial plasmids, which are circular DNA structures using the enzyme DNA ligase. You start off with your circular double-stranded plasmid DNA. That's going to be your cloning vector. We break it, we cut it using the restriction enzyme that we are working with. We also cut the DNA from the fragment that we want to clone, either from an organism or from a cell, or could be even, any of those things. And you are then going to attach it into the plasmid, right? You create this essentially a reaction mix that contains the clone fragment to be cloned, the plasmid DNA, your cloning vector that's already cut with the restriction enzyme, and then you allow for them to work together to create that covalent linkage with DNA ligase, leading to your recombinant DNA molecule. So this is your recombinant DNA molecule, and this is your cloning vector. And this will now contain this new gene or this new DNA sequence that we are now going to be able to study or work with. Once you have created your cloned DNA fragment inside a vector, once you've placed it in there, you want to have lots and lots of copies of it typically to work with, so that you can do the work downstream. This usually requires that we introduce this particular vector inside a bacterial cell, and we do that by transforming our cells, right, with this particular double-stranded recombinant plasmid DNA. And once it's inside the bacteria, we can culture it, just like any other cultures that we make of bacterial cells. Typically, the vector that we are using will contain some type of a selection marker. So maybe it also contains, in addition to our DNA sequence of interest, it also maybe contains an ampicillin resistance gene or some other resistant gene, so that we can grow the cells in the presence of that particular antibiotic or the selection. So we have a selection media, selective media to grow these cells, and only cells that actually got the plasmid will grow. And so as they grow, you are producing now many millions of copies of that particular bacteria, and thus of the plasmid. Once we have grown it in the log phase, right, so you let it grow, so there are lots and lots of these bacteria there, and then we collect it and we extract the plasmid from these bacterial cells by lysing them open and purifying that DNA fragment. Okay, so once we have our DNA, we can then double check for whether it was indeed cloned properly or not. For example, using, again, gel electrophoresis, we can reuse the same enzyme that we used before to create the cuts again. This time you would then have fragment with the black bone and then fragment that was excised out. Then you can run it on an agarose gel with a current running through it. DNA, because of the phosphate in the backbone, is negatively charged, so it will automatically go from negative to positive. And you are able to then examine it to see how many fragments are there, how far they traveled. If you use a ladder with known size fragments of DNA, you can compare your fragments against it to see if they were of the right size or right place, if you got what you expected to get. So here's the cool part about this. Using similar mechanisms, we can actually create what we call genomic libraries, which contain DNA fragments that represent the entire genome of that particular organism. Basically, what we do to create something like this is cut up the entire genomic DNA for the organism using a restriction enzyme. And then once you have basically cleaved the entire genome into lots and lots of small pieces, you clone those fragments, those individual fragments that have been made, into plasmids using DNA ligase. So each one of these fragments, whether or not it clones for an actual gene, will get inserted into plasmids, and you will have a large library of bacteria, each one containing its own little fragment of this human DNA, for example. So these would be your recombinant DNA molecules. Now, in this example, I'm just showing these colored portion, but it doesn't have to be just that. It's going to basically take up everything. Once that has happened, you can then basically look at all of them, have this entire set of bacteria, each one containing a portion of the genomic DNA in them, and freeze it until you need it. And this would be your genomic library that we can use then later on to sequence individual portions and to create overlapping DNA sequences that can help us sequence the entire genome for that particular organism. Another thing we can do with this, obviously, is not just looking at genomic DNA, which would be a lot of DNA, right? So if you remember with humans, majority of our DNA is non-coding DNA. And so copying every single last portion of that into a genomic library seems like a little bit overkill if you're interested in actual function of genes and function of specific structures. So instead of doing the entire genomic library, we could say, hey, I only care about the portion of the genome that is active in my favorite cell. I study hepatocytes or liver cells, and I wanna, or I study. I'm studying what is activated in livers, and so I'm just gonna look at hepatocytes and that is liver cells, and look at what are exactly the genes that are active in those cells, only in liver cells. Similarly, I can say, hey, I don't really care about livers or any other part of the body. I only care about the neurons, or I only care about astrocytes in the brain that create astrocytoma. So I care about what different genes are active inside just the astrocytes. In that case, you don't want to actually create a genomic DNA library, but instead you want to do what we call complementary DNA, or cDNA, which is essentially prepared from mRNA of the genes that are active in a particular cell. So in a complementary DNA library, instead of taking the genomic DNA, you first use a reverse transcriptase to go from mRNA, the messenger RNA, back into a DNA form. That is what we call the complementary DNA, or cDNA. And then you take those cDNA fragments and you create libraries of only the expressed portion, basically. Only the proteins that are gonna be actively produced inside those cells, only. And this way you can examine portions of genome that are active under very specific conditions, whether in a particular cell type, in a particular developmental time frame, under disease, phenotype, whatever the case may be. So to do that, basically we take the cells that are being cultured, or we may take a tissue or a portion of the cells from the organism themselves. We lyse the cells and we extract all the messenger RNA from them. Once we have the messenger RNA, we make a DNA copy using a reverse transcriptase to go from RNA to DNA, and then we use the reverse transcriptase not only to form that initial RNA DNA structure, right? RNA DNA hybrid double helix, but also we can partially degrade that RNA with RNase and synthesize a complementary DNA strand to it using DNA polymerase. This leads to creating a new molecule structure that is basically the double-stranded complementary DNA, or cDNA molecule, which can then be input into a library, again using the same technique as before. This obviously helps us then create a big library that contains just the information that we need for our particular cell type about which proteins are being expressed or which genes are actively expressed in those particular cells. Now, knowing that some sequence exists, or not even some sequence, that some portion of the DNA is actually actively being copied in RNA and used for protein is one thing. The next thing you want to know is, well, what exactly is the sequence that is being copied and that is being utilized for protein production? To do that, we are going to look at multiple methods for that as well. So one of the things that we do, once we have a DNA molecule that is of interest, once we have isolated it, we can separate the two strands of DNA, and then we can look at individual single nucleotides in each of those strands and look at their actual sequence and use that to utilize that to figure out what exact sequence is present there. You can because DNA, again, a lot of it is made with just hydrogen bonds, bond type of associations, you can actually heat the DNA, and if you heat it high enough, you can eventually melt it or denature it so that you have single strands in there, because the hydrogen bonds would be broken at that time. If you want to renature it, you can slowly cool it down and allow for those hydrogen bonds to be restored, and that would give you the same sequence back again. This hybridization can occur between, obviously, DNA double strands, but also you can have it between complementary strands of DNA and RNA molecules, or two RNA molecules as well. And so using that method, we can create structures that can be then studied further for sequence analysis. So just kind of going, doing a little review of this. When we are isolating and cloning DNA molecule, there are lots of different tools that researchers have. We used restriction enzymes isolated many times from bacteria to cut DNA molecules at specific sites. We can then utilize gel electrophoresis with agarose gels to separate the DNA fragments based on size as they move through charge field, going from negative to positive. DNA cloning begins with production of a recombinant DNA molecule, typically in a cloning vector, a backbone that is a circular plasmid in many times. In many cases, these recombinant DNA molecules that are basically a circular plasmid with the insert of your favored DNA, of your cloned DNA present inside it, can be copied inside bacterial cells to create large amounts of this particular cloned recombinant DNA, which can then be purified and used for whatever purpose we need. You can also utilize similar technique to create entire genome libraries for different organisms by cutting the entire genome with particular enzymes and then cloning each fragment that you receive into individual cloning vectors. In addition, you can have hybridization of DNA and RNA, or two different nucleotide molecules, when the sequence is the same or very similar, so that you can examine how closely related the two different sequences are. Okay, so the next part is basically, okay, so we got our recombinant DNA. Now what? How do we clone this DNA into another place? So one way that we can get the fragment of interest is obviously going straight to the organism and cutting it out. But remember, genomes are very complex, and genomes have lots of different places where the same restriction enzyme may be cutting. So if we were just making a stab in the dark, we are going to end up with all kinds of different molecules and not just the one that we cared about. So instead of this, if we know the sequence of a particular DNA, or if we have a particular fragment that we know contains our sequence of interest, even if we do not ourselves know the exact sequence of the in its entirety, as long as we have some information about the sequence, we can use a technique called polymerase chain reaction, or PCR. PCR is an in vitro technique where basically we amplify or copy many times a portion of the DNA that lies within two known sequences. So you don't have to know everything in that sequence, as long as you know kind of flagging portions of that particular area that we are interested in. As long as we have some information about, you know, just kind of ends of it or some portion of it, we can fill in the blanks using PCR, and then we can take it out and sequence that particular DNA. So PCR amplification requires that we know something about the sequence surrounding that area that we are interested in, even if we don't know the entire sequence of that region itself, because we are going to be utilizing complementary sequences to those outer regions, to those regions of interest, which are going to be composed of oligonucleotide primers. We will call them primers. So remember, when we are doing just simple transcription or replication, the DNA polymerase require a primer as well, right? It's complementary to the place that it's starting at. And that's what it hangs on to and works with. Well, in this case, we are going to provide it the primer by telling it where to go, right? Because the base sequence, that's why we need to know that sequence, at least on the outside, because we are going to make this little tiny primer that will act as a little arrow and tell it, here is where you start. And then the second one that tells it, here is where you stop. So these primers are typically short nucleotide sequences. These are single-stranded, which are complementary to the region that we are trying to get to, or outer regions of those sequences, right? And they are usually anywhere from 16 to 24 nucleotide pairs. These oligonucleotides serve as primers for, again, the DNA polymerase, and they are going to bind to those sequences, the complementary sequences, once the genome has denatured and you have single strands as templates. And then they can be utilized just like you would in a transcription or in a replication setting, so that you can get a new strand of DNA which is complementary to your parent template strand. They will again work 5' to 3' just like we did before. So the 5' end is, again, it's going to go from 5' to 3', right? And so we want to know at least one side. You don't have to know both sides. You have to at least know one side, where to start. And then from that point onward, you can just leave it open-ended so that it kind of just goes as far as it goes and falls off, and then you get different fragments of different lengths, and then sequence each one of them and kind of use the overlapping portions to align them. Or you can know both sides of it. You know that there is a gene somewhere in this 3000 base pair area, and so you create a primer to the beginning of that portion where you know already have that information about, and something at the end, and then you copy the region in between. In another scenario, maybe you already know the sequence of the gene itself and you are interested in cloning that particular gene of interest for whatever purpose, to create a resistant crop or to create a molecule or an organism with a particular characteristic. In that case, you could just take the beginning and the end of that gene and actually use those as your primers so that you can copy the entire sequence that goes in between. So here is an example for that. So you have basically your primers that are going to be primer complementary to your coding strand, which is the strand that you care about, and complementary to the non-coding strand. And the region in between it is what's going to be copied to get you your final sequence that you are going to be cloning. And in this particular technique, in the first portion, you're going to heat the DNA so that it separates, and you're going to heat those primers at the same time so they separate and you have single-strand DNA structures. Those single-strand DNA structures are then cooled slowly so that the primers can anneal to their own complementary region on those two strands. We then provide it with DNA polymerase and all the nucleotides, right? So ATP, GTP, CTP, and TTP, so that they can fill in the gap as they read, and they can fill in those nucleotide sequences. And so at the end of the first cycle, you're now going to have two molecules where that first strand, right, is again the entire genome strand, but the second one, the complementary to that, is that has just been built by your pair of primers. It's going to be starting at the right place, but then ending wherever it feels like. Similarly, in your second strand, the same thing would have happened. It's going to start at the complementary non-coding strand primer and then go on until it falls off. So in the second cycle of amplification, it will now again separate out, and each one of these strands are going to get their primers attached to them and then continue. And so in the end of the second cycle, you're going to have 4 molecules, 2 of which are going to look just like this, and the other 2 are now going to have the correct sequence that we were looking for. So here you can see that example. You have the product from the first cycle. In the second strand, you repeat this process, and now you have 2 that again are going to have 1 strand that is going to start at the correct place, and the second one will be the genomic one, so it will have the entire sequence. And then when you continue it another time, a third time, in that third cycle, you finally produce your actual DNA fragments with the correct length on both sides. So now you are going to end up with these molecules that are, you'll have 8 total molecules, out of which you will have several that are going to be the correct size and the correct sequence. So for example, this one is going to give you the correct size and sequence. In this case, this one's going to give you the correct size and sequence. So it's the third cycle that will give you the correct size and sequence for the first time, and it's going to give you 2 molecules like that. And then it will continue on in each subsequent turn until you get to, typically we do around 30 cycles. And that will give you a lot of 30 to 40 cycles to get to a lot of copies of your desired DNA. PCR can obviously be used to obtain either clones from DNA, from genomic DNA, or from cDNA libraries as well. But remember, if it's the genomic DNA, your clone is going to contain a lot of non-coding information as well. But if it is a cDNA clone, it's going to contain just the coding portion of your genes, and it will be kind of simpler in its size and complexity. So here you can kind of see that example. So in here you have your entire DNA, you're the chromosomes, you have a DNA segment that we want to clone, and that's going to get cloned in this case, and it will contain both coding and non-coding information. However, if you were looking at isolating the mRNA specifically and then cloning the portion that we need, you're just going to get the coding portion of those particular genes. This can be used to detect presence of viral genomes in samples of blood. So we can use this particular technique not only for cloning and for creating GMOs or to get a particular function from these cells, but also to identify infectious particles in our systems. PCR is also used in forensics, where we can use it to distinguish different people from each other through analysis of specific portions of our genome that are non-coding but important in their own way. So we look at, and we talked a little bit about this last time, that there are these small tandem repeats inside our sequences, and there are lots and lots of them. So we can use these repeats and look at the number of repeats that are present in a particular individual and match it with our victim or culprit and see if it matches, because that is something that is highly variable. These short tandem repeats, or STRs, are extremely variable in population and go from anywhere from 4 to 40 in different individuals. So here in this particular example, we have 3 different individuals that are our suspects, and then we have our forensic sample. So you want to find out which one of these 3 individuals is most likely to be the culprit. They should match all 3 of these STR sites exactly the way they are. So looking at just this, right, these are just giving you the number of repeats that are present in at those STR sites, at those 3 STR sites, both for both of the chromosomes, or for both of the allele sites, right, both sides of the DNA, one for maternal and one for paternal. And so we can look at them and we can see which ones match up. To match up, every single fragment has to be there in both, and that's the case for individual B. Individual B matches the forensic sample. You can look at it here as well, and you can also look at the banding pattern and see that they too have exactly the same sequence. So DNA can be cloned. Specific portions of DNA can be cloned using PCR. PCR is basically polymerase chain reaction. It uses DNA polymerase and specific DNA primers that we design to amplify sequences that we desire in a test tube, and it is extremely important in diagnostic and forensic applications. Okay, so the last topic that we'll talk about today, actually in this lecture, was sequencing DNA, and then for the second half of the chapter 10, we will I will post another lecture at another time later on. So to sequence the DNA, we use a specific method that is called the dideoxy method of sequencing. It relies on specific nucleotides that are made that cannot be extended any further. So they are basically, they terminate the chain once they're incorporated into the molecule. So in this case, we set up the sequencing reaction so that they have a lot of the normal nucleosides, nucleoside triphosphates, and then a small tiny amount of deoxy triphosphates, DDNTPs. The DDNTPs, they prevent the strand from getting extended at the 3' hydrogen instead of the hydroxyl, which allows for the extension. So they basically have a 3' hydrogen instead of hydroxyl, so they cannot be extended. And so some small portion of the time, if it only takes like 1%, then 1% of the time, this is the one that's going to be picked up randomly by the reaction. And so sometimes it's going to be the very first nucleoside that's taken and it stops. Sometimes it's the second, sometimes it's the third. So you will have a range of fragments in your final reaction product that has every different combination. And so you can run it on the gel and basically read from bottom up and see exactly what the sequence is. Read it up. So the normal deoxyribonucleoside triphosphate has the hydroxyl. The chain terminating dideoxyribonucleoside DDNTPs has a hydrogen that prevents strand extension at the 3' end. So here is an example of what happens when this reaction occurs. You have your single-stranded DNA fragment that we are trying to sequence. You add your primer against it so that you can copy it, start copying it. You have lots and lots of unlabeled normal deoxy-DNTPs, and you have a small amount of these labeled dideoxy nucleotides. And then each time it's going to have different place that it picks up this molecule so that it stops at different time. Now each one of these are fluorescently labeled with a different color so that when we separate them in our gel at the end, and these can be loaded onto a special capillary gel, so it's a very thin, long gel, and you can read off based on the fluorescence that comes off. So in this case, you can see it's TTC C, T, A, T, right? So you just kind of read based off of the color that's coming at each point, from the shortest to the highest, to the longest. This is most of the times this part is done through computer softwares that can read it much easier and much quicker than we can. Now, these type of things can help us sequence the entire genome as well. Again, this can be done using a combination of methods. So one of these methods is called shotgun sequencing. This is usually what we use, shotgun sequencing, for tiny or small, relatively small genomes. In this, the genome is first broken up into many species using, you can use different restriction enzymes or whatever you want to create small overlapping sequences. And then you are going to sequence each one of those fragments. And because you have these overlapping ends, you're going to be able to line them up so that you can assemble that final genome once all is said and done. So that's what you are doing here. You have your entire genome, you cut it up, and you cut it up with different enzymes so that you have overlapping sequences in different tubes, and then you sequence each one of these fragments, you line them up based on how they line up with each other, and assemble your final sequence. Now, this is great in a typical small genome, but a genome like a human that have a lot of repetitive DNA sequences, it's not going to work very well, is it? Because those repetitive DNA sequences are going to make it very difficult to find the correct overlapping ends to line up. And that way you can result in sequences where you have two different fragments from different parts of the DNA. They may look like they're overlapping, but they are not really in the right position if you put them that way. And that can give you incorrect final assembly. So in that case, anything that was in between those two very different sequences is going to be lost in the process, and you will have to figure it out at a later time. So in that case, you can do some other types of methods as well. So one of those is using what we call back clones that can be used to position, that can be positioned all along the genome and then used to create a sequence based off the restriction site signature. In this, the clones are essentially digested with 5 different restriction enzymes. And you look at the overlapping portions based off of that, right? Because you have these different enzymes that are cutting the same portion, right? One segment. And you can look at the restriction pattern for individual back clones within a sequence. And this will give you a lot more distinctive pattern that can then help you line them up. Again, the second portion is the same. You're going to sequence every single one of these at the end of the day so that you can line them up and line up all the restriction sites, and then that will allow you to organize them in a better, more complete, more accurate fashion. So we use these techniques all the time in our labs in order to correctly sequence a genome, to identify organisms, to identify what is present inside them. Now we also use this type of method to examine genomes as well, and to sequence them with the help of computers and computer programs. So one of the ways that we can do large-scale sequencing is using a method called Illumina sequencing. This is basically using the dideoxy sequencing method, but in this case you take a large genome that is then cut up into millions of short fragments, lots and lots. So you cut them up into a lot more short fragments than before, each one containing a small sequence. You attach each one of these fragments to a glass surface. So you have this particular slide that has lots and lots of spots, may have thousands of spots. Each one is going to contain a particular sequence that is then going to be amplified by PCR using random primers to create DNA clusters. Each will contain, anywhere from a few hundred to 1,000 copies of that particular DNA molecule that's stuck to that particular spot on the slide. Each one of these are then, all these anchor DNA clusters are then incubated with DNA polymerase, and again, a set of 4 nucleotides like we did before. And some of them are going to be, again, the uniquely colored fluorescent that don't allow for further addition, and some that will still allow for it. And then we are going to be using this. So this is the fluorescent tag and terminator in here that we are going to be using to repeat the process, and again, sequence it as we go. And in this way, you can get a snapshot of each round of synthesis. At the end of each PCR cycle, essentially, you are going to get a snapshot of it and looking at what is tagged at the end at that time. And then it's going to do it again, and then take another picture, and then do it again. And so for each cycle, you're going to get the next nucleotide that is present there. And that's going to allow you to get the sequence for that particular area. Now, we use these type of methods to do not only sequencing of a single organism, but also comparative genome analysis, where we can identify different genes, line them up on the genome across various organisms, and predict what their function might be based on the different portions, the different domains that are present within them, the genetic differences that are happening from one species to another, and so on and so forth. So, to sequence the DNA, just to recap, the main method of choice is dideoxy sequencing. That depends on analysis of DNA chains terminated at every position using these dideoxy nucleoside triphosphates that don't allow for further addition to their chain. Next generation sequencing techniques allows us to do this a lot cheaper and a lot faster using computer programs, so that we can get these done in a more timely manner. And we can use these methods to compare genomes across different organisms to predict and identify genes and their function. So next time we are going to be talking about exploring gene function. So we got the genes, we got them sequenced, so now we're going to talk about how we would examine their function in the next lecture. Until then, take care. Bye-bye.